there! Ready for a wellness adventure? Let's explore the world of health together!
Have you ever wondered why proteins are considered essential for life, yet the body can make some of their building blocks, but not others? To answer that question, we first need to understand what proteins are actually made of, and how the body builds them every single day. Proteins are large biological molecules constructed from smaller units called amino acids, which are often described as the building blocks of proteins. Although thousands of different proteins exist in the human body, they are all assembled from the same 20 standard amino acids used in different combinations. You can think of amino acids as individual letters of an alphabet, while proteins are complete words and sentences created by arranging those letters differently. A small change in the order of amino acids can completely change the shape and function of a protein inside the human body. Some proteins act as enzymes, others provide structure, some transport molecules, and many serve as hormones or components of the immune system. Every muscle contraction, immune response, wound healing process, and cellular repair mechanism depends on proteins made from amino acids. Unlike carbohydrates and fats, the body does not maintain a large storage reserve of amino acids specifically for future protein production. Instead, amino acids are continuously recycled from existing proteins and obtained from dietary sources to support ongoing physiological functions. The 20 standard amino acids are divided into two major categories called essential amino acids and non-essential amino acids. The word essential in nutrition does not mean more important or biologically superior than non-essential amino acids. Instead, essential means that the human body cannot produce enough of that amino acid to meet physiological requirements under normal conditions. Because the body cannot synthesize adequate amounts, essential amino acids must be obtained through food consumed as part of the diet. Non-essential amino acids receive their name because the body can synthesize them internally from other molecules through normal metabolic pathways. This does not mean non-essential amino acids are unnecessary because they remain essential for normal physiology and protein production throughout the body. The difference simply reflects where the amino acid comes from rather than its biological importance for maintaining health and cellular function. The 9 essential amino acids for humans are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Each essential amino acid participates in protein synthesis and contributes to multiple physiological processes depending on where proteins are needed. Leucine, isoleucine, and valine are sometimes grouped together because their chemical structures share similarities and influence muscle metabolism. Tryptophan serves as a precursor for serotonin, which functions as a neurotransmitter involved in communication within the nervous system. Phenylalanine contributes to the production of tyrosine, which can participate in the synthesis of several important signaling molecules. Methionine contains sulfur and contributes to metabolic pathways involved in the production of various cellular compounds and proteins. Lysine plays important roles in collagen production, which supports connective tissues, including skin, tendons, ligaments, and bones throughout the body. Histidine contributes to the production of histamine, a signaling molecule involved in immune responses and several physiological regulatory mechanisms. Threonine participates in protein synthesis and contributes to structural proteins found in connective tissues and intestinal protective barriers. The body requires all essential amino acids because protein synthesis cannot proceed efficiently if one required amino acid becomes unavailable. Imagine building a bicycle using a kit that contains every necessary part except for the chain connecting the pedals to the wheel. Even if every other component is available, the bicycle cannot function properly because one essential part is missing completely. Protein synthesis follows a similar principle because the absence of one essential amino acid limits the production of complete functional proteins. This concept is sometimes referred to as the limiting amino acid principle in nutritional science and protein metabolism studies. If one essential amino acid is present in insufficient amounts, overall protein synthesis may become constrained despite adequate availability of other amino acids. Now let's look at non-essential amino acids and how the body produces them internally through normal metabolic processes. Examples of non-essential amino acids include alanine, asparagine, aspartic acid, glutamic acid, and serine, among several others. The body synthesizes these amino acids by modifying compounds produced during carbohydrate metabolism, fat metabolism, and protein metabolism pathways. For example, glucose metabolism generates intermediate molecules that can serve as starting materials for non-essential amino acid production. This metabolic flexibility allows the body to adapt to changing nutrient availability while maintaining essential physiological processes. Some amino acids occupy an intermediate category called conditionally essential amino acids under certain physiological circumstances. Normally, the body can synthesize these amino acids in adequate amounts for routine maintenance and everyday metabolic functions. However, during periods of rapid growth, severe illness, injury, burns, infection, or intense physiological stress, production may become insufficient. Under these conditions, additional dietary intake may help meet increased physiological demand while supporting tissue repair and recovery processes. Examples of conditionally essential amino acids include glutamine, arginine, cysteine, tyrosine, glycine, and proline, depending on physiological circumstances. Glutamine serves as an energy source for certain immune cells and cells lining the intestinal tract under many normal conditions. Arginine participates in the production of nitric oxide, a signaling molecule involved in blood vessel relaxation and vascular regulation. Tyrosine can be synthesized from phenylalanine, but may become conditionally essential if phenylalanine metabolism becomes impaired. The body uses amino acids for much more than building skeletal muscle, despite the common association between protein and athletic performance. Enzymes responsible for digestion, metabolism, and cellular chemistry are proteins assembled from amino acids obtained through dietary intake and recycling. Hemoglobin, the oxygen-carrying molecule inside red blood cells, contains protein components built from amino acids, continuously synthesized by the body. Many hormones depend on amino acids either directly or indirectly for their synthesis and structural organization within endocrine tissues. Antibodies produced by the immune system are specialized proteins designed to recognize and bind specific foreign substances entering the body. Collagen, the most abundant protein in the human body, provides structural support for skin, cartilage, bones, tendons, and ligaments. Keratin forms the structural framework of hair and nails, while contributing to protective barriers within various tissues throughout the body. Transport proteins move nutrients, minerals, hormones, and other molecules through the bloodstream and across cellular membranes continuously. Muscle tissue itself consists largely of proteins called actin and myosin that interact during contraction to produce movement. Because proteins perform so many functions, amino acids are constantly needed for maintenance, repair, and synthesis throughout the entire body. Dietary proteins are first broken down during digestion before their amino acids become available for absorption and subsequent protein synthesis. Protein digestion begins in the stomach, where hydrochloric acid unfolds protein structures, making them more accessible to digestive enzymes. An enzyme called pepsin starts breaking proteins into smaller fragments known as peptides during the digestive process. Digestion continues in the small intestine, where pancreatic enzymes further break peptides into individual amino acids and smaller peptide fragments. These amino acids are absorbed through intestinal cells and transported into the bloodstream, where they become available for tissue use. The liver acts as an important processing center, helping regulate amino acid distribution according to physiological needs throughout the body. Some amino acids are immediately incorporated into newly synthesized proteins, while others temporarily circulate, awaiting metabolic demand. Unlike fat storage or glycogen storage, there is no specialized amino acid storage organ reserved exclusively for future protein synthesis. This means dietary protein intake and continuous protein recycling become important for maintaining amino acid availability over time. When energy intake becomes insufficient, amino acids may also contribute to glucose production through metabolic pathways in the liver. However, amino acids are not the body's preferred energy source because proteins serve numerous structural and functional roles. Breaking down large amounts of protein for energy may reduce availability for tissue repair and normal physiological maintenance activities. Physical activity influences amino acid metabolism because exercise increases both protein breakdown and protein synthesis within muscle tissue. After exercise, amino acids contribute to repairing microscopic structural changes produced during normal muscle contraction and adaptation processes. Growth during childhood and adolescence increases protein requirements because expanding tissues require continuous synthesis of new structural proteins. Pregnancy also increases amino acid demand because maternal tissues and developing fetal tissues both require ongoing protein production simultaneously. Wound healing depends heavily on amino acid availability because damaged tissues must produce collagen and many other structural proteins. The immune system increases protein synthesis during infections because antibodies and immune signaling molecules require amino acid building blocks. Even ordinary daily activities involve constant protein remodeling because cells continuously replace damaged or aging proteins throughout the body. The body carefully regulates amino acid metabolism because both deficiency and excessive imbalance may disrupt normal physiological function. Understanding essential and non-essential amino acids helps explain why proteins are far more than simply nutrients associated with muscle development. Amino acids support virtually every organ system through their roles in enzymes, hormones, structural proteins, transport molecules, and immune defenses. In summary, proteins are built from 20 amino acids that combine in different sequences to produce thousands of functional biological molecules. Essential amino acids must come from food because the body cannot produce sufficient amounts internally under normal physiological conditions. Non-essential amino acids can be synthesized by the body, while some become conditionally essential during growth, illness, injury, or increased metabolic stress. Together, these amino acids provide the raw materials required for building, maintaining, repairing, and regulating the countless proteins that sustain human life. Have you ever wondered why your body needs protein every day if your muscles and organs are already made of protein? A common misconception is that once proteins are built, they remain unchanged for the rest of our lives without requiring replacement or maintenance. In reality, the human body is constantly breaking down old proteins and building new ones through a process called protein turnover. Protein turnover refers to the continuous cycle of protein degradation and protein synthesis occurring in virtually every tissue throughout the body. This process allows damaged proteins to be removed and replaced while helping tissues adapt to growth, stress, injury, and changing physiological demands. You can think of the body as a city where buildings are constantly being repaired instead of waiting until they completely collapse. Roads develop cracks, bridges require maintenance, and damaged structures are repaired before they become unsafe for public use. Similarly, cells continuously inspect proteins for wear and damage, replacing them before their function becomes significantly impaired. This constant renewal helps maintain normal physiological function despite ongoing exposure to mechanical stress and chemical reactions. Protein turnover begins at the cellular level because proteins experience gradual damage during normal metabolic activity throughout everyday life. Heat, oxidation, mechanical movement, and ordinary chemical reactions can gradually alter protein structure and reduce functional efficiency. Cells possess quality control systems capable of identifying proteins that are damaged, misfolded, or no longer functioning correctly. Once identified, these proteins can be broken down into amino acids and recycled for the synthesis of entirely new proteins. This recycling process helps conserve resources because amino acids, recovered from old proteins, remain valuable building blocks for future protein production. The body carefully balances protein breakdown and protein synthesis according to physiological conditions and metabolic needs. If protein synthesis exceeds protein breakdown, tissues may increase their protein content over time through gradual accumulation. If protein breakdown exceeds synthesis for prolonged periods, tissues may lose protein mass and structural integrity gradually. Muscle tissue provides one of the most familiar examples of protein turnover because muscles continuously adapt to physical activity and inactivity. Muscles are largely composed of contractile proteins called actin and myosin that generate force during movement and physical exercise. Even during complete rest, muscle proteins are continuously broken down and rebuilt as part of normal maintenance and repair. During exercise, especially resistance training, muscle fibers experience microscopic structural stress resulting from repeated contractions under mechanical load. These small structural changes are a normal consequence of muscle function rather than evidence of harmful injury or permanent tissue damage after exercise. Muscle cells activate protein synthesis pathways that repair existing structures and replace damaged contractile proteins gradually. Amino acids absorbed from food and recycled from older proteins contribute to rebuilding muscle tissue during this recovery process. However, Muscle protein turnover does not occur exclusively in athletes because ordinary daily movement also requires ongoing maintenance and repair. Walking, climbing stairs, maintaining posture, and performing household activities all contribute to normal muscle protein remodeling over time. Periods of inactivity also influence muscle protein turnover, because reduced mechanical stimulation alters the balance between synthesis and breakdown. Extended bed rest, prolonged immobilization, or reduced physical activity may increase muscle protein loss through altered metabolic signaling. The body adjusts protein metabolism according to physiological demand because maintaining unnecessary tissue requires energy and amino acid resources continuously. Now let's examine enzymes, another major category of proteins undergoing continuous turnover throughout the human body. Enzymes are specialized proteins that accelerate chemical reactions without being permanently changed or consumed by those reactions themselves. Every digestive process, metabolic pathway, and cellular repair mechanism depends on thousands of enzymes functioning continuously throughout the body. Although enzymes can catalyze many reactions repeatedly, they eventually experience structural damage and require replacement through normal protein turnover. Digestive enzymes produced by the pancreas and digestive tract are synthesized continuously because they are secreted and eventually removed from the body. Metabolic enzymes inside cells also undergo turnover, because ongoing chemical activity gradually affects their structure and functional efficiency. The liver continuously synthesizes numerous enzymes responsible for metabolism, detoxification, nutrient processing, and production of important biological molecules. The digestive system adjusts enzyme production according to physiological conditions and nutrient availability through regulated protein synthesis pathways. This adaptability allows the body to respond efficiently to change in nutritional conditions and metabolic demands across different situations. Now consider the immune system, which depends heavily on protein turnover for effective defense against infectious agents and environmental threats. Antibodies are specialized proteins produced by immune cells that recognize and bind specific foreign substances entering the human body. When the immune system encounters unfamiliar microorganisms, it increases antibody production through activation of specialized immune cells. These antibodies are proteins assembled from amino acids, obtained through dietary intake and recycling of existing protein structures. Immune cells themselves contain thousands of proteins that require ongoing synthesis, maintenance, and replacement throughout their functional lifespan. Cytokines are signaling proteins that allow immune cells to communicate and coordinate responses during infection and tissue repair processes. Complement proteins circulate in the bloodstream and participate in identifying and eliminating potentially harmful microorganisms entering the body. Because immune proteins are continuously synthesized and degraded, protein turnover remains important even in individuals who appear completely healthy. Infections and injuries can increase protein turnover because immune activity and tissue repair require additional protein synthesis simultaneously. Wound healing provides another example of extensive protein turnover occurring in response to physiological demand and structural tissue damage. When skin is injured, the body increases production of collagen and numerous other structural proteins involved in rebuilding damaged tissues. Fibroblasts produce collagen fibers that strengthen the healing area while supporting the gradual restoration of normal tissue structure. Blood clotting also depends on proteins synthesized primarily by the liver and activated rapidly when blood vessel injury occurs. Many hormones or proteins or peptides that undergo continuous synthesis according to physiological needs and regulatory feedback mechanisms. Insulin, for example, is a protein hormone produced by specialized pancreatic cells involved in blood glucose regulation. Transport proteins also experience turnover, because molecules responsible for carrying nutrients and hormones require periodic replacement and maintenance. Albumin, produced by the liver, transports various substances through the bloodstream and contributes to fluid balance regulation. Hemoglobin inside red blood cells contains protein components responsible for transporting oxygen from the lungs to body tissues. Red blood cells themselves have limited lifespans and are continuously replaced through protein synthesis occurring in the bone marrow. The body regulates protein turnover carefully because excessive protein degradation could impair tissue function while inadequate degradation allows damaged proteins to accumulate. Cells possess specialized systems for degrading proteins that are old, damaged, or no longer needed for normal physiological function. One major pathway involves the proteasome, a cellular structure capable of breaking proteins into smaller peptide fragments and amino acids. Another important maintenance system is autophagy, which allows cells to recycle damaged proteins and even defective organelles under certain conditions. Autophagy helps maintain cellular quality control while recovering valuable molecular components for future biological use and synthesis. Nutrition influences protein turnover because amino acid availability affects the body's capacity for synthesizing new proteins efficiently. Dietary proteins provide amino acids that support replacement of muscle proteins, enzymes, immune molecules, and structural proteins throughout the body. Energy availability also affects turnover because protein synthesis requires ATP generated through carbohydrate and fat metabolism within cells. During prolonged energy deficiency, the body may increase protein breakdown to provide amino acids for glucose production and essential metabolic functions. This adaptation helps maintain blood glucose supply for tissues that depend heavily on glucose as an energy source. Growth during childhood requires elevated protein turnover because expanding tissues must continuously synthesize new structural and functional proteins. Pregnancy increases protein synthesis because maternal tissues and developing fetal tissues require simultaneous growth and maintenance processes. Aging influences protein turnover because synthesis and degradation rates may change gradually over time across different tissues and organs. Research from aging institutions indicates that older adults may experience reduced responsiveness of muscle protein synthesis under certain physiological conditions. Physical activity helps stimulate muscle protein synthesis and supports ongoing maintenance of skeletal muscle tissue throughout adulthood. Sleep also contributes to protein turnover because many repair and growth processes become more active during normal sleep cycles. Growth hormone secretion during deep sleep supports tissue maintenance and protein synthesis in several physiological systems. Protein turnover demonstrates that the human body is not a static structure but a continuously adapting biological system requiring constant maintenance. Old proteins are removed, amino acids are recycled, and new proteins are synthesized according to changing physiological demands and environmental conditions. In summary, protein turnover is a continuous cycle of protein breakdown and protein synthesis occurring throughout the entire human body. Muscle proteins, enzymes, immune proteins, transport proteins, and structural proteins all undergo constant renewal to maintain physiological function. This ongoing process allows tissues to repair damage, adapt to activity, support immune responses, and maintain the countless protein-dependent functions required for human life. Have you ever wondered why some proteins are called complete while others are described as incomplete? And what those terms actually mean biologically? Many people hear these terms and assume that complete proteins are automatically healthy, while incomplete proteins are somehow inferior or useless. In reality, the concepts of protein quality and completeness are much more about amino acid composition than about whether a food is good or bad. To understand protein quality, we first need to remember that proteins are built from 20 standard amino acids used throughout the human body. 9 of these amino acids are considered essential because the body cannot produce enough of them to meet normal physiological requirements. Since essential amino acids must come from food, the amino acid composition of dietary proteins becomes an important nutritional consideration. A complete protein contains all 9 essential amino acids in amounts sufficient to support normal human physiological functions and protein synthesis. An incomplete protein contains 1 or more essential amino acids in relatively smaller amounts compared with physiological requirements for protein production. The word incomplete does not mean that a protein lacks all essential amino acids or that it has no nutritional value whatsoever. Many incomplete proteins contain every essential amino acid but have relatively low amounts of 1 or more compared with the body's needs. The essential amino acid present in the smallest relative amount is often called the limiting amino acid in nutritional science. Imagine trying to assemble a bicycle using 100 screws, 100 bolts, and only 20 wheels available for construction. Even though most parts are abundant, production becomes limited by the component available in the smallest required quantity for assembly. Protein synthesis follows a similar biological principle because all essential amino acids must be available for efficient protein construction. If one essential amino acid becomes insufficient, protein synthesis may slow despite adequate availability of the remaining amino acids. This concept explains why amino acid balance is important when considering overall dietary protein quality and physiological requirements. Animal-derived foods such as eggs, dairy products, fish, poultry, and meat generally contain complete protein profiles naturally. These foods typically provide all essential amino acids in proportions that support human protein synthesis and tissue maintenance efficiently. However, complete proteins are not limited to animal sources because certain plant foods also contain all essential amino acids. Soybeans, quinoa, buckwheat, and a few other plant foods provide relatively complete amino acid profiles suitable for supporting protein synthesis. Many plant foods, however, contain lower amounts of one or more essential amino acids relative to human physiological requirements. For example, grains often contain relatively lower amounts of lysine compared with several other essential amino acids naturally present. Legumes frequently contain relatively lower amounts of methionine, while providing substantial quantities of lysine and many other amino acids. This difference in amino acid composition led scientists to develop the concept of complementary proteins many decades ago. Complementary proteins are foods with different amino acid patterns that together provide a more balanced essential amino acid profile. A classic example involves grains and legumes because their limiting amino acids differ from one another nutritionally. Grains provide relatively more methionine while legumes provide relatively more lysine, creating complementary amino acid patterns when consumed regularly. Importantly, complementary proteins do not necessarily need to be consumed during the exact same meal to support physiological protein requirements. The body maintains circulating amino acid pools that allow amino acids from different meals to contribute to protein synthesis throughout the day. This means dietary variety over time contributes significantly to amino acid availability and overall protein quality within normal eating patterns. Protein quality depends not only on amino acid composition but also on digestibility and amino acid absorption efficiency. Digestibility refers to how effectively digestive enzymes break proteins into absorbable amino acids and small peptide fragments. Some proteins are digested rapidly, while others require longer processing within the digestive tract before absorption occurs. The digestive process begins in the stomach, where hydrochloric acid unfolds protein structures, making them more accessible to digestive enzymes. Pepsin begins protein breakdown in the stomach before pancreatic enzymes continue digestion within the small intestine progressively. Amino acids and small peptides are absorbed through intestinal cells and transported into circulation for use throughout the body. If digestion and absorption are inefficient, fewer amino acids become available for protein synthesis despite adequate dietary protein intake. Researchers have developed methods to estimate protein quality by considering both amino acid composition and digestibility characteristics. These assessment methods help scientists compare different dietary proteins according to physiological amino acid availability after digestion. However, no single numerical score fully captures the complexity of human nutrition because dietary patterns involve many interacting variables. Protein quality should therefore be viewed as one component of overall nutritional physiology rather than an isolated characteristic. The body continuously mixes amino acids obtained from various foods with amino acids recycled from existing body proteins every day. This recycling system provides additional flexibility because amino acids released during protein turnover can contribute to future protein synthesis. The liver plays a major role in amino acid metabolism by processing absorbed amino acids and regulating distribution to various tissues. Muscle tissue, immune cells, connective tissues, and internal organs all compete for amino acids according to physiological needs. Growth during childhood increases demand for complete amino acid availability because expanding tissues require continuous protein synthesis. Pregnancy also increases amino acid requirements because maternal and fetal tissues simultaneously depend on protein production for growth. Wound healing requires adequate amino acid availability because collagen and many structural proteins must be synthesized during tissue repair. The immune system depends heavily on balanced amino acid supplies because antibodies and signaling proteins require ongoing synthesis continuously. Physical activity influences protein quality considerations because exercise stimulates muscle protein turnover and tissue remodeling processes. After exercise, amino acids support repair and replacement of contractile proteins damaged during normal muscle activity and adaptation. Energy intake also influences amino acid utilization because adequate energy helps preserve amino acids for structural and functional purposes. If energy intake becomes insufficient, amino acids may increasingly contribute to glucose production and other metabolic energy pathways. This shift demonstrates that amino acids serve multiple physiological functions beyond simply building muscle tissue throughout the body. Protein quality also changes depending on the overall dietary context rather than any individual food consumed in complete isolation. For example, meals containing multiple protein sources may provide broader amino acid distributions than meals relying on a single source. Whole foods containing protein often provide additional nutrients, including vitamins, minerals, fiber, and healthy fats, supporting overall physiology. Legumes provide protein, along with fiber and minerals, while dairy products provide protein together with calcium and other nutrients. Nuts and seeds contribute protein, as well as unsaturated fats, vitamin E, magnesium, and numerous plant compounds simultaneously. Fish provides protein, along with essential fatty acids, that contribute to membrane structure and signaling functions within tissues. This illustrates that protein-containing foods contribute multiple nutrients, interacting together within normal human physiology and metabolism. A common misconception suggests that excess dietary protein can be stored in the body similarly to glycogen or body fat reserves. Unlike glycogen and triglycerides, the body does not maintain a dedicated storage organ specifically for excess amino acids. Unused amino acids may be incorporated into proteins, converted into other molecules, or metabolized depending on physiological circumstances. Protein requirements therefore reflect ongoing synthesis, repair, maintenance, growth, and adaptation rather than accumulation in specialized storage tissues. Understanding protein quality helps explain why amino acid composition and digestibility influence physiological protein availability across different dietary patterns. Complete proteins provide all essential amino acids in appropriate proportions, while incomplete proteins may contain limiting amino acids requiring dietary variety. Complementary proteins demonstrate that different foods can work together to improve overall amino acid balance within regular eating habits. The body further supports this flexibility through amino acid recycling and continuous protein turnover occurring throughout every organ system. In summary, protein quality refers to amino acid composition and digestibility, while completeness refers to the presence of all essential amino acids. Complete proteins provide adequate amounts of essential amino acids, while incomplete proteins may contain one or more limiting amino acids. Dietary variety, complementary protein sources, digestion, absorption, and protein recycling all contribute to maintaining the amino acid supply required for normal human physiology. If this content helped you better understand proteins and amino acids, consider subscribing to the channel to continue exploring the science of nutrition and the human body in the next chapters of this educational series. Thanks for watching World Wellness, Health, and Nutrition.
Thanks for watching! Hope you liked it!